Hello friends, I welcome you all to my another video tutorial of a Biotech guide. And in this video lecture, we are going to discuss about regulation of a gene expression. Okay, so here you focused gene expression. So what is gene expression? As we know, in human, basically a process where the protein is formed. So how protein is formed, and what is the mechanism behind that? Central dogma, where what happened? Formation of the mRNA is defining as a transcription, and after that protein is synthesized, is defining as a translation. Okay, and whole process is basically happening on one mechanism, that is a gene expression. If gene is not expressed, so mRNA is not formed, and if mRNA is not formed, protein is not synthesized. Okay, so basically gene expression in human, a mechanism is there is defining as a central dogma. But how is happening in the bacteria? So for that a mechanism is there, and this mechanism is defining as a operon. Operon is nothing but is a operating unit. Okay, and this operon is classified into two: inducer and a repressor. So in inducer type of operon, example is Lac Operon. Three structural genes are there and basically happening in the E. coli. Likewise, same in repressor, trip operon. Here trip is defining as a tryptophan operon, and 5 structural genes are there, and this is also happening in the E. coli. Brief we are going to discuss about in this video lecture is Lac Operon. Okay, so as usual, today's video going on a very interesting level. Stay tuned with us and enjoy this video lecture. So let us start first with introduction. Definition: what is Lac Operon? So as we discussed, Lac Operon is designated as a Lac Operon which codes for an enzyme involved in the catabolism or a degradation of a lactose, is defining as a Lac Operon. Okay, here Lac is stand as a lactose sugar, and Operon is stand as a operating unit, and also called as a clusters of gene. Well, brief discussed in next. And then who described first? So Jacob and Monod in 1961 at the Pasteur Institute Paris described this Lac Operon. And also we discussed that is defining as an inducible operator system, because this is the type of a induced type of a operon. Let us discuss structure of Lac Operon. So basically you can see here 4 elements are there. This is element 1, 2, 3, and this is the 4th, which is defining as a structural gene. Okay, here I is defining as a regulatory gene, then P is defining as a promoter, O defining as a operator, and after these 3, Lac Z, Lac Y, and Lac A, these are the genes also defining as a structural genes and also known as a polycystonic gene. Okay, so this is the structural 4th element of a Lac Operon. Now we discuss this 4 element function. So first, Lac I doing what? Controls the production of a repressor protein. As we know, the function of repressor protein is what? To inhibit or stop the reaction. Okay, so that reaction stop via this Lac I element. Then second, Lac P defining as a promoter, and this is the place where the RNA polymerase bind. So this is defining as a binding site of RNA polymerase. Then third, Lac O is a binding site of a repressor. Okay, brief mechanism we'll discuss in next. Before that, focus on these all function. Okay, then third, structural genes. We discuss Lac Z, Y, and A. These are the structural genes. Then these genes are codes for this enzyme, and this is the function of this enzyme. Okay, so first, Lac Z gene is codes for the beta galactosidase enzyme, and function of beta galactosidase is what? Breakdown of a lactose into a glucose and a galactose. Then Lac Y is codes for the permease enzyme, and the function is provides entry for a lactose in the cell. As the name suggests, permease give a permission. Then Lac A is codes for two enzymes, trans-acetylase and a thiogalactoside. So trans-acetylase doing what? Adds acetyl group to beta-galactosidase to activate. Okay, here beta-galactosidase enzyme needs activator, and this activator is acetyl, and that acetyl is provided by this enzyme trans-acetylase. Then another enzyme is thiogalactoside, which having a unknown function. Okay, so this is the all about of this element function. Now let us discuss function of Lac Operon. For that we have to assume two conditions. First is absence of lactose or inducer, and second presence of lactose or inducer. Here lactose is defining as a inducer. So first, when lactose is absent, so what happen here? Here regulatory gene produce a protein that is known as a repressor protein, and that repressor protein binds with this operator region. Okay, so here repressor protein is formed, and that repressor protein is bind with this operator region. In result, what happen? These all enzymes these all structural enzymes do not work and don't transcribing mRNA, and if these structural genes are not in working condition, mRNA is not transcribing. So in result, protein means what? These enzymes, these genes producing enzyme like beta galactosidase permease, as we discussed previous, these all enzymes are not produced, and in result catabolism or a degradation of a lactose is not formed. But second scenario, if lactose is present, so what happen? A small amount of a sugar that is defining as a allolactose. Okay, this allolactose sugar is formed within a cell, and this bind with this repressor protein. Okay, so here this allolactose sugar is bind with this active site of a repressor protein, and resulting reduce the affinity. Okay, reduce the affinity. How? By changing the conformational change of this repressor protein, and what happen? No longer bind with this operator region, and at the end result, this repressor protein is now released. Okay, this repressor protein is now released, and further RNA polymerase is now bind with this promoter region, and in result these all structural genes are in working condition. Now this RNA polymerase is permit to these structural genes for producing mRNA synthesis and further their protein or an enzyme. Now let us go through theory. So for that we have to assume four conditions. When lactose is absent, also we discussed, and when lactose is present, which is also we discussed. But now another two condition, both lactose and glucose is present, so what happen? And when glucose is absent and lactose is present, so what happen? So first, when lactose is absent, repressor protein formed by regulatory chain and continuously synthesized and bind with the operator site, resulting is what? No mRNA synthesis. Then this repressor protein blocks the promoter site, which is the binding site of RNA polymerase, where the RNA polymerase started transcribing. Okay, so here when lactose is absent, no more mRNA synthesis there, and result no protein production. Second condition, when lactose is present, so as we discussed, a small amount of a sugar allolactose is formed. This feeds on the repressor protein. Basically active site of protein, repressor protein, there is also defining as a allosteric site, where this allolactose bind with this repressor protein, and this causes the repressor protein to change its shape or conformational change, and result into no longer bind with the operator site, and resulting RNA polymerase is now reached on a promoter site, and formation of a mRNA and further transcribing process. Now another exciting condition is when glucose and lactose is present. So what happen? When the glucose and lactose are present, so RNA polymerase can shift on the promoter site but it is unstable and keeps falling off. So here when both sugar are present, like glucose and lactose, so the promoter region RNA polymerase is bind, but it is in unstable condition and keeps falling off. Last four condition, when glucose is absent and lactose is present, so what happen? Another protein is needed for this condition, defining as activator protein, which binds with the RNA polymerase and stabilizes it. Okay, and this is the only condition when this activator protein is synthesized. Which condition? Glucose is absent and lactose is present. And this is the how E. coli only makes enzyme to metabolize other sugars. So here we completed our video. I hope you enjoying very well and got it very well. Thanks for watching, and if you want to get more and more updates regarding like this, so join with us, subscribe to our channel, press the bell icon, never miss an update. Thank you so much friends.